Any questions, concerns so far? We have, yeah, it's handwritten. So we'll print out the exams and we'll bring them to the class and you'll basically write your answers and then we'll collect the exams. They'll be graded and then you'll have your exams the next week, the following week. So the exam should not take more than an hour or so. But you'll have the whole class. We'll put out the study guide, and the study guide, as I said, it will be a bunch of essay question type. And you'll have that, and you can study it. Most of the short essay questions on the exam will be from that study guide, but there will be some short, like multiple choice questions that will be from the lecture slides, but not necessarily from the study guide. So that is to encourage you to study the whole slides and the material that we covered and not just the study guide. The study guide is mostly just for the short essay questions that will come on the exam. We'll publish, I'll put it up maybe tomorrow, the study guide, and then we'll have a session. The TAs will run a session this Friday from 10 a.m. to 12 p.m. to noon. That's the Zoom meeting for it. You have access to it, so you should all attend that. If you cannot attend it, they're going to record the session and they'll make the recording available. You should try to come to that meeting prepared. You should study a little, just find out where your weakest spots are. Come to that meeting with an understanding of your weaknesses. And then you could ask them about it, and then they'll be happy to help you. It's a 2-hour slot, so there's a lot of time to cover a lot of material. Yeah, we'll have the exam next week. Then later next week, we'll have a couple of tutorials from Olivia and Nadia on how to read scientific papers, how to write a paper in APA-Style. You'll need those two for the assignment that's be coming up, which is due early October. So you should also all plan on attending these tutorials. Olivia will get the information in here, but this stuff for Nadia's tutorial already in there. Questions? Yes, you all feel confident then? So in terms of the material and stuff, we're not really covering anything too complicated, I think, yet. We're kind of, these are the basics and foundational stuff. So it's good that you have a good understanding of these material before we go to some more complex stuff. All right.
All right. I actually got a question about the essay questions, and that is this is not a full essay that you're writing, but short answers, right? So
yeah,
several sentences, but it's not the kind of essay that has like a beginning, middle, and an end. It's just short answers.
Yeah, short essay questions. They're not to a few minutes, right? Yeah. What's that?
One paragraph. Sentences.
Depends on the question. Some of them may be longer. You'll write as much as it's necessary to answer the question. Okay, perfect. So last couple of weeks we talked about neurons. We talked about how they communicate, how they influence one another. Last time we talked about how you could abstract those properties and build something that people call artificial neural networks, because they're not natural, they're artificial, they're built in a computer and they mimic some properties of the natural neural networks that we have in our brains. that was an abstraction. That was something that people have built. They can do stuff. They do stuff. They're in our, on the internet. A lot of people use them. They're in our phones. Um, Today, we're going to talk about some variations of natural neural networks, the way people have used these ideas to understand what happens in the brain. Before that, I have a sentence here. I think it's an important premise, something again that you should internalize as uh, as an important uh, foundation to cognitive neuroscience. That that all of our behavior, perceptions, and cognition um, can be ultimately reduced to a complex pattern of activation within a population of neurons in the brain. What does that mean? Well, any emotional state that we have, any cognitive or perceptual experience that we have, any actions that we have, these are all eventually have to be some activity in the brain, a coherent, concerted activity of many neurons in the brain in order for that thing to happen. Um, so you put the plastic bag away? No, no. Someone don't make that noise with plastic. Um, so we're gonna look at some of the um, behavior that is with the vision, and we're gonna use a network that is much simpler, right? Um, it's not in the scope of this course to look at really large networks, but we're going to look at much simpler networks in order to just understand the concepts. Now imagine this network. This is a network that it starts from photoreceptors. We'll talk about photoreceptors more down the line. We'll talk about the anatomy of the eye. We'll talk about how does the eye convert light into neural activity, right? That has to happen initially. When there's light hitting your eye, or when you hear a sound on your ear, that energy in the physical world has to be converted into neural activity. And we'll talk about how that happens for different modalities. But imagine in this abstract way, we have a bunch of photoreceptors. And these photoreceptors get light on them. And that light activates these photoreceptors. They become active. They activate neurons that are connected to those photoreceptors. And those neurons could activate other neurons that are connected to these neurons, and so on. So it's a very abstracted network. Now, in this schematic, I'm going to use this shape to identify an excitatory interaction, and this shape to identify an inhibitory interaction. So it means when you see this, it means that photoreceptor 1 excites neuron A. Photoreceptor 7 excites neuron C. Neuron C basically excites neuron B, right? So, and you all know what that means. It means that if A's activity goes up, if A's firing rate goes up, as a result, the B's firing rate will go up. So if I put light, if there is a light that is hitting all of these photoreceptors, you would imagine that the neuron B would be much more active than if the light only hits photoreceptor 4, right? This is what I'm trying to show here. If more receptors are stimulated here, the result of that on neuron B is increased firing rate, right? Well, let's look at here. We're saying, what if light only hits 4? There's light there, and then there's some activity for neuron B. What if light then hits 3 through 5? So 3, 4, 5. Why should the activity here be for neuron B would be higher than when the light only hits 4? Okay, so yes, so there's more neuron, there are more photoreceptors that are active, and all of them have excitatory connection to B. So if more of them are active, then they would activate B more because that's an excitatory connection. Now, if you expand your light and then hit 6 and 7 and then or 2 through 6, well, if 2 gets light, then A is going to be activated because this is excitatory. A is going to in turn excite B because this is excitatory. So if 2 through 6 is active, if the neuron B would have a higher firing rate than only 3 through 5. If 1 through 7 are active, then it would be the highest, right? That this neuron cannot possibly have a higher firing rate than in this case, from 1 through 7. Again, for the purpose of this course, imagine these are photoreceptors, these are inner neurons in the eye, and then these are ganglion cells. These are all pretty much in the retina. We'll talk about them later. Now let's take another example. If 4 is activated, as I'm showing here, right? These are the receptors that are stimulated. You would have a certain firing rate for neuron B. What happens when you activate through 3 through 5? Would the activity of neuron B change? Why wouldn't it change?
Because it's not receiving any stimulus.
Exactly. Right? So B is not connected to 3 and 5, right? This is equivalent to having a 0 connection in our neural network. If B is not connected to 3 or 5, then if you put light, if you expand your light, no matter how much you expand your light, it's not going to change the activity of B because B only is responding to the activity of 4. So the concept that we're gonna use is that sometimes there is convergence, that a lot of photoreceptors converge into a cell. Sometimes there is no convergence or very little convergence, that not that many photoreceptors converge into a cell. So the effect of that is, well, one of these is going to have a much bigger so-called receptive field, and we're going to talk about that. So the receptive field is really the region that a cell responds to. Now, it's easy to think about this in the context of vision because there's light in the environment, and then you could say, well, if I flash a light here or flash a light there, I could see where in this environment the neuron responds to, right? Again, with this schematic that we had earlier, if you put light at A, at position 1 here, well, B gonna respond to it. If you put at position like 4, like imagine a light moving in the environment, and then if you hit photoreceptor 7, well, it's gonna respond too. If you hit all of them, it's all gonna respond to. But if you hit the light now to this photoreceptor, what is the impact of that on the activity of B? What is it? Nothing. So it means that these photoreceptors are outside, like the light that is coming is outside the receptive field of this cell. That's what it means. Like this cell only responds when the light is hitting these photoreceptors. You could do this now with a cat, right? Imagine that was in a schematic, but you could do this in a cat. And I was saying a cat is sitting there watching the screen, and you have a pattern of light. You could move the light in the environment. You could move the light on the screen and then see at what point this cell responds. You're just listening to one cell. And this is neurophysiology, and you've all seen what neurophysiology means by now. You could move this light on the screen. You can make it bigger, make it smaller. The electrode is inserted into the visual cortex of a cat, which is in the back of the head, as you could see here. And I said, just coarsely know the anatomy of the brain. And as the light moves around, you just hear the cell. And we're going to hear it in a little bit as I play it. Then you could see, well, in certain area of on the visual environment, the cell is going crazy. Like it's firing really hard. And then you basically move it around in that area, And then you draw a circle, say, look at this. This is the receptive field of this cell. That this is specific cell in the brain only cares about visual input coming from this part of the screen or this part of the visual world. A lot of this is influenced by this research. And again, I played this a little bit to introduce you to neurophysiology, but we're going to watch it a little bit more. So the sound you hear are the firing rates, basically the firing of a cell that are hooked up to an amplifier and then to a speaker. You're hearing the cell, and that's a good thing, right? That way you can actually know what you're doing. You can see when there is light in this specific area of a space, this cell is firing really hard. You could hear it, right? When the light comes on, you could hear the cell firing harder, right? When the light goes off, then you can't hear it. So there's an excitatory area. There's an area of the visual world that light in that area excites the cell. But imagine this is a network, right? This is not, the cell is not sitting right in the eye, but rather a multi-layer network that gives the cell this sort of so-called selectivity. So that's the process that I described. You move the light around, to play around with it, and then see which part of the environment really activates this. You can see something that we're going to talk about is that a cell usually has an excitatory area and a lot of time has an inhibitory area. That light in an area in the visual world can really inhibit this cell. So a cell also basically combines all these excitatory and inhibitory inputs in order to have some baseline activity or above or below that baseline. So again, think of it as that gas area or a brake area basically for this cell. Here you can see when the light narrows down to just that specific area that is excitatory, the cell would fire more. Right? You all see that? You can hear that. All right, so we'll watch more of that, but let's get back to this. So this is the process that you'll find where the receptive field of a cell is. This is the area in the space that can actually impact this cell. Now, how does a cell get this sort of properties, right? How does a cell in the brain, what defines its properties? What defines any cell's properties? What is it? What defines its behavior? What? Its charge? It's, yeah, but what leads a cell to have a certain kind of selectivity? Meaning that what makes a cell, what makes a neuron in the brain to only be responsive to this part of this space, for example? What's that? The potential, no, so you're talking about the behavior of a cell on a micro scale. I'm talking about a cell has a certain job, sometimes you could say. Its job in this case is to detect light in this part of the space. And that is because So what does it give that receptive field properties?
Function.
Okay,
A change
change.
in the receptive field.
Yeah, but what does it, how does a cell gets to have certain receptive field properties and another cell different receptive field properties? What is it in a network, as I talked about, where is all this information stored? In the connections, right? The way a cell is connected defines its function, defines its receptive field, defines all its properties. There are two things in the brain. There are connections and there is activity. Okay, everything has to reduce to these two, or that's the premise of cognitive neuroscience. It could be all wrong, but what the premise that we work with is that there are connections and there are activations. So any cell, here's an example. What makes this cell to have a large receptive field? It responds to a lot of area in the visual space, and these cells to have a small receptive field. What does that? What does that? The connectivity, right? This cell is connected to a lot of photoreceptors. That cell is connected to only one photoreceptor in one part of the space. So its connectivity defines its function. And cells have very different connectivity. Yes.
I mean, wouldn't its function be just defined by like how when you're born, just certain cells are intended to do certain things?
Intended as because they're connected in a certain way.
I guess I don't think that the connection has a consciousness like or like certain neurons just are. That's what they're born to do, right?
Sure. So certain cells. So it's a complex story, right? So there are cells that starting from a single cell, right? Uh, it grows and it divides into more cells and more cells, and some cells take the function of the heart, some cells, take the function of the lung, some cells eventually become brain cells, right? Um, but in order for a function to emerge, you have to be connected in a certain way, right? If you're not, so what makes, so for example, I say, well, the area in the back of the brain is visual area. Why is it the visual area? Why isn't the visual area in the front of the brain? Why is that?
Because the neurons, the back of the brain are connected to photoreceptors.
Exactly, right? Because the eye, the connections from the eye go there. So the connectivity overrules everything. If you're connected to the eye, you're going to be overrun by visual input. So you become visual input cells. That's what I mean. I don't know if there's a deeper problem you have with this, but we could address that. Here in this case, right, one cell's properties are defined by their connectivity. If it's connected to more receptive fields or more cells that are responding to light, like photoreceptors, then it would have larger receptive field. So another way to say that this cell has a larger convergence compared to these cells. They don't have large convergence. So in a lot of cases, the receptive field of earlier areas that are closer to the photoreceptors are circular in shape. And you saw example of that in that video. So, and then, as I said, the size of the receptive field generally increases with convergence. If there are areas that have higher convergence, and we'll talk about this later. For example, when you go from central vision to peripheral vision, right? We all have central vision and peripheral vision. We'll talk about that later. In the peripheral vision, you have higher convergence. So you have bigger receptive field for areas that are farther from your fixation point. And a lot of time, the organization of these receptive fields are center surround. And that means that the responses are not the way I showed earlier, right? In this example, it's all excitatory. everything, every photoreceptor here eventually has an excitatory effect on neuron B, right? And any photoreceptor that you activate, yeah. Which circle? Where my mouse is?
Yeah.
That's just the unit representing the cell body. Yeah, you could say, call it cell body. No, these are, so A, B, and C are different neurons. 1, 2, 3 through 7 are photoreceptors. Sorry, I thought this was clear. I should have been more clear then. So A, B, C are, so everyone understands it? A, B, C are different neurons. This is their axon. So this is the axon from A to B, and it's excitatory. Axon from C to B is excitatory. These are photoreceptors, and those are also cells. And the photoreceptor axons to, for example, cell B here are excitatory. 6 and 7, excitatory to C. These are cells, and these are photoreceptors. So here, that's when I say receptor stimulated, is basically how much light we're shining here. So are we just shining light on receptor 4? Are we shining a bigger light? And I showed you in the video, you could really play around with these lights, make them bigger, make them smaller, move them around. And that's the idea that you see in the graph. That here, it's all excitatory. Any receptor that you activate here, it's going to excite cell B eventually. Does that make sense? You all see this, right? This is very basic. You need to understand before we move forward. Any photoreceptor here activated will excite cell B eventually. Yeah, they're on the back of the eye. Yeah, we'll talk about them later. Yeah. We'll talk about the anatomy of the eye later. Yeah.
So all the photoreceptors in this diagram are excitatory. There's no inhibitory ones.
Photoreceptors cannot be excitatory or inhibitory per se.
Oh, okay.
Photoreceptor gets light, it gets excited.
Yeah. I guess
The connection to
are all excitatory.
Yes.
Okay. Perfect.
You have to look at the connection. What is the connection between a photoreceptor and a cell? What is the connection between a cell and another cell? Yeah. The lines, which line?
Just clarifying that that's like a structural representation, not like a sense. Yeah. Oh, like the neural network, not the neural. Correct. But is it a two-way, like that little line underneath? Like, I don't know.
Are you saying that this line that my mouse is on?
No, go higher.
This one?
Yeah, go.
These are photoreceptors and they're connected to cells. So a photoreceptor is excitatory connected to this cell. So it's and this is a cell itself. So this is a presynaptic cell, a body of a cell or a photoreceptor, and that's excitatory connected to cell A. So this is a presynaptic cell, and A is the postsynaptic cell. And when 2 gets light, it excites A. So the firing rate of A goes up. Make sense? So here, I'm using this graphics. If this is the shape which looks like a stop sign, this is inhibitory. And if it looks like this small triangular shape or a pigeon feet, that's excitatory. This is a schematic, right? So I'm showing a network as a schematic, not all the detail, not all the anatomical details. I'm saying this is a network, and given that I'm using this terminology, then these are excitatory connections. Make sense?
So on the
slide where you have the output of B with that, on the slide that has with conversions and without, the with conversion roughly like represent peripheral vision and without convergence.
No, so you have a lot of
I mean,
convergence.
it's showing a network of like an input and then what's the output? What you see?
At this point, we're just talking about the activity of the cells. So we're just saying, if this is the input pattern of light, what would be the activity of this cell? It doesn't have to be a percept at this point. Yeah.
Some cells have this behavior, like you said, like you have in this drawing here on the top, and what differentiates?
So some cells in the visual cortex have a connectivity that has a high convergence. Some cells have low convergence. The good thing about low convergence is that you're not averaging out a lot of responses, meaning that, so if there's a small amount of light and there's another light next to it, if you have a high convergence, you don't see it as two different lights. That's why in the peripheral vision, if I'm looking at my hand in the peripheral, like there's low resolution, right? Because you're not really differentiating between all the little details. You have to look at it to differentiate. And that's why, as I said, we have low convergence in central vision, but high convergence in peripheral vision. It's a trade-off. You can't have low convergence everywhere because then you have to pack a lot more cells, and you can't. So the brain finds this, or evolutionarily, we have this trade-off where you have really good central vision and good enough peripheral vision. That's the idea.
So for the cells that we talked about briefly in the last class that fire based on like orientation of a line of light, would it look something like that, but maybe like 1 and 2 would be specifically from just the orientation versus like 3 through 7 would just be the light itself, or is there?
No, we'll talk about those. I know what you mean, but we'll talk about it. So at the end of this, if we get there, we'll talk about it. So what we're looking at here is a cell B has a receptive field that is purely excitatory. Do you all understand what that means? It has a receptive field that is purely excitatory. There is no part of the visual world that if you put a light, it's going to inhibit this cell. Yeah? But that's not usually what happens. What really happens are where were we? What really happens is something that, so this is the example that I showed you, that in this case, it's all excitatory, but what really happens is that you usually have what we call center-surround organization. That in the center, for example, is excitatory and the surround is inhibitory. Or the opposite, in the center is inhibitory and surround is excitatory. Well, what would that look like? Well, look at this case. If I have this connectivity, if I have this network, and I'm only looking at the activity of cell B, for example. Well, look at cell B, and let's say and this is our electrode that we managed to put in the brain and just touch cell B and record its activity. If I put light on only on only the receptor, photoreceptor 4, I'm going to get an activity. What happens if you just expand our light like those experimenters in the video did to hit 3, 4, 5? What happens to the cell activity? Does it go up or does it go down? Go up. Why? Yeah, because 3 and 5 are also excitatory. So if you hit all 3 of them, it's all excitatory. Now, what if we expand it further? What if we expand it to 2 and 6? What's going to happen to the activity of the cell? What's that? It's going to stay the same. Why? So let's look at it. If I shine light on 2, what's going to happen to the activity of A? In this case, there's no light on 2. Let's say I expand my light and light now hits the 2. What happens to the activity of A? It goes up. If A goes up, what happens to the activity of B? What's this connection? Okay. So if A goes up, what happens to the activity of B? It goes down. So if I look at 6 again, if also the light expanded enough to hit 6, what happens to the activity of C? What happens to the activity of C? It goes up. What happens to B? It goes down. So starting from this point, if the light expands to hit 2 and 6, what happens to the activity of B at the end? It goes down. It has to go down because when you were hitting 3 through 5, it was all excitatory. Now you expand your light enough that you hit some of the inhibitory, some of the brakes. So it brings down the cell. What happens with the now expand the light further to hit 1 through 7? What happens to the activity of the cell? What happens? Why does it go down? Yeah, so now if I light up 1, like what happens to the activity of A from this point on, where the light is hitting 2? If I hit the light to 1, what happens to the activity of A? It goes up because these are both excitatory to A. So if there's more light, the activity of A goes up. What happens to the activity of B? It goes down. Same happens on this side. So if it's 1 through 7, would the activity of B be higher than this condition that was only 4 or be lower? What's that? Why would it be lower than even this case?
Because there's more inhibitory than excited.
Yeah, so if you excite 1 through 7, you have 1 and 2 that are inhibitory eventually for B. 6, and I say eventually, right? This is a network. There are multiple layers. You could be exciting some cells, and then these cells exciting somebody else, and that's inhibiting somebody else. So the cascade is eventually inhibitory. At the end, you'll have 4 inhibitory photoreceptors and 3 excitatory. So you'll even go below where you were when you hit 4. Does that make sense to everyone? You have to follow this. This is going to be on the exam. You're going to get a network, and I'm going to give you a pattern of light and ask you if the activity is going to go higher or lower. So you have to understand what this network does. Now, given the behavior of this cell, you could say, well, the cell has an excitatory center and an inhibitory surround, right? So the center is excitatory and the surround is inhibitory. Does that make sense to everyone? Yes, let's just start from this side. What's that?
Do you mind?
So this is the center, 3 through 5. And 6 and 7 are the surround. That's just the simple center surround. And if the center gets light, this cell, what happens to this cell? What happens to the activity of cell? Excites. Okay, this is 1 through 7. What happens if I turn off 4? That there is no light in 4, so I'm just giving it the donut light pattern. What happens to the activity of B? Does it go lower than this or higher than this? Lower. Why? You have 1 less excitatory input. So if you give it a donut shape, it's hitting even more inhibitory versus excitatory. If I turn off 1, what happens to the activity of C? If I turn off 1, what happens to the activity of C? Nothing. It's not connected. Okay? Which one of these cells have the biggest receptive field? B. I think you'll understand it. That's good. All right. This is excitatory center because, well, the center has excitatory connections. Inhibitory surround because the surround has eventually inhibitory for B. But you can understand that the receptive field of B and C are different. The receptive field of C is all excitatory. The receptive field of B is excitatory center, inhibitory surround. Let's see. So when they narrow the light and the cell activity goes up, what does it mean? Is this what is the property of the receptive field here? What is it? It's excitatory center then, right? If I narrow the light, as you could hear here, if I narrow the light and the cell activity goes up, it means that the center is really excitatory and the surround is inhibitory. That's why when there is no light in the surround, or the light in the surround gets smaller, the cell activity goes up. And that means that it's excitatory center, inhibitory surround.
Is there just no sound for inhibitory, or is it just lower sound than excitatory?
It's lower sound. So it's harder. Yeah, it's not as easy as, oh, the cell goes completely quiet or something like that. It's just that it's the frequency of firing is lower.
Quieter, the sound is further apart.
Yes. So so what we're hearing is the firing rate. So every dot is a firing. So the frequency of firing. But because if the cell is really active, the sounds mix up and then you hear it as a louder thing, but it's not really louder. It's just that they overlap. It seems because the sound is slower than the firing rate, so you perceive it as being louder. So what we saw now are the on center. An on center is another way of saying excitatory center. You could say on center of surround, right? That means exactly excitatory center, inhibitory surround. So that's another terminology. But you could also have cells that are the opposite, that are off center and on surround, meaning that a donut shape light would excite the cell more because it's not hitting any light in the center. But you should all be now able to imagining what's going on.
Sorry, I don't know if anybody.
Just put it down, that's fine.
I got it.
So you should all now be able to imagine what does it mean if I told you draw a network where the cell is excitatory surround, inhibitory center, you should be able to imagine what a network looks like. Right? You should be able to imagine, okay, if I connect it in this way, any pattern, if I tell you, well, this is the pattern that this cell responds to, you should be able to imagine that network. Yes. Well, but I have to give you a pattern of light.
You give me patterns, like, okay, I
Yeah, yeah. Yeah, the cell by itself would not be enough, right? You have to have a pattern of light.
you would have to show me that it's either hitting the center.
Yeah. This is a cell that they're trying to map out, right? This is the process, and it's important for you to understand the process of mapping out a cell. This happens a lot in every, neurophysiology or any other experiment. You really have to understand, like even if you're studying faces, for example, well, you have to show faces and just understand which part of the brain is responding in order for you to go and put an electrode there. It's the any cognitive neuroscience experiment goes through this process. So now you can see, like prior to this, it wasn't responding as strongly, but when they have this donut pattern, you could see that it's stronger because now you're not hitting the inhibitory center, but you're trying to hit the excitatory surround. And that way you're trying to find, so this is a process of finding a lot of cognitive neuroscience is the process of finding what is the best stimulus, or stimulus being an input. What is the best stimulus for this specific brain area or this specific cell? And that's a lot of cognitive neuroscience. And this is the process, basically. As I said, you could do it with a lot of different things. You could do it with, emotional faces. You could do it with many different stimuli. It could be a pattern of sound. But a lot of the experiments are trying in this process to find what is the right stimulus for the cell that I'm recording from. Yes.
And this, in order to determine all this, it's like the single cell, it has to be invasive.
Yeah.
So this is only done with animals, right?
As I said, there are cases. What did I say? I said there are cases you could do this in humans. Yeah, but most of the research obviously is done in animals. But as I said, you could have fMRI stuff that are not done in animals, right? You're recording from the whole brain, and you could show different stimuli, and you're interested in a very specific area in the brain. You could see if the fMRI response goes up or down. If you show a face and show it in different areas of the brain, different areas of the screen, and show it with different emotional valence, and see what happens with this cell. But in this case, because they're studying the visual cortex and they're studying early visual areas, these are areas that are really close to the eye, close, not distance-wise necessarily, but in terms of the number of layers that you go from the photoreceptor to the cell. That's what I mean by closeness. Finding these cells are harder. Like quantifying their responses are harder. The on center, off surround one are much easier to find. All right, let's take a 10-minute break and then we'll continue. So, what, as I said, what we call this sort of receptive field are basically center surround cells. Their receptive field is roughly circular, as you see here, and there's an antagonism between the center and the surround. In this case, as I said, it could be, and again, this is another way to display a receptive field. This is an abstraction. Just try to, this is not like a reality, right? This is just an abstraction of, if I want to communicate the behavior of this specific cell that is down here, right? Every cell here would have a very different behavior. But if I wanted to communicate the behavior of this specific cell, then I would show it like this. Which means that if there is light in the center, it's going to excite this cell. And if there is light in the surround, it's going to inhibit this cell. So it's inhibitory or off surround and excitatory on surround, because, well, if it's in the center, it's going to turn on this cell in a certain way. And if it's in the surround, it's going to turn off this cell. Now, you could imagine, as I said, an opposite organization. You could imagine an inhibitory center and excitatory surround. How would this look like here? How would this connectivity be different for a cell, for this specific cell to have that receptive field? Well, that would mean that the stuff that are coming from the center would have to inhibit the cell. That means that, well, the stuff that are coming from the center, what is the impact on this cell? Excitatory. But this cell in this network is exciting this cell, but this could have been an inhibitory. This could be a line here and inhibit this cell. Alternatively, these two could be excitatory. So you could see with just 3 changes here, I could change the receptive field to be inhibitory center and excitatory surround. Right? You could all see that network. Yeah.
Just want to clarify, this is happening at a really tiny scale. This is for like one cell. This is not a receptive field for like an entire
Just
eye.
one cell. Just one cell. So as you in the video, we're just listening to one cell.
So there's like thousands of this happening.
Or like millions of, yeah. So if you have this receptive field in the world, right, in the screen, you could say, okay, this cell is responding to this part of the world. There is a cell next to it that responds to similar. And then if you move away a little, the receptive field is going to move a little, right? So that's called a retinotopic organization, and we're going to talk about that. Questions? Yeah.
2 and 5 and 6 get light, and 3 and 5 don't. What would that look like?
Receptive, okay. Receptive field is a property of the connectivity, right? Receptive field has nothing to do with the light pattern itself. I'm summarizing this network in this receptive field. I try to, right? There are 2 differences here. Like anatomy has certain properties, connectivity has certain properties. This receptive field only summarizes the connectivity of this cell. Does that make sense to everyone? It summarizes the connectivity. It says that if there were light in this part of the world, it would excite this cell. If there is light in this part of the world, it would inhibit this cell. Now, then you could decide, well, with this light pattern, what would be the activity of the cell? So dissociate these two. There's anatomy and there's activity, yes.
I have a question about just that experiment we've been watching with the light cat and electrode. Is the electrode stimulating the cat's brain and then the cat is seeing?
No.
It's the opposite.
Yeah, there's no stimulation.
Okay, so the electrode is just measuring.
It's just recording.
Got it.
Yeah. The electrode is just listening into what this cell is doing. The activation is all driven by the light. Yeah.
In terms of function, why would it be beneficial in some cases for the receptive field to have excitatory center and then an inhibitory surround, or the opposite, inhibitory center excited? What is the functionality of those?
Well, the functionality is that, as we'll see, this is a network. You build on the previous layer. If you have these excitatory center, inhibitory surround, and inhibitory center, excitatory, then you could detect edges with them at a different layer. Then you could, with those edges, you could detect corners. With those, you could detect, shapes. With those, you could detect objects. So it's a hierarchy, and at every level of the hierarchy, you basically detect certain things. Right? We're here, let's say we're looking at 3 levels of this hierarchy: photoreceptors, inner neurons, ganglion cells. Then you see what I mean? It's important for the network to have access to cells that are responding differently in order to detect edges.
Could you not achieve the same thing if you had no convergence with each photoreceptor connecting to a visual cell? Or would that be too much?
Well, no, you can't. So any important pattern in the world would be a collection, right? It wouldn't be just one dot in the world. Yeah. Every if I want to detect a face, I have to detect it in a certain area. So a lot of cells have to respond together across many layers in order for me to perceive a face. So convergence is necessary for a system. For any hierarchical system to have any behavior, there has to be convergence.
This visualization on the right here, is that's because there's more inhibitory cells for the middle cell here and less excitatory?
No. So this is just summarizing the cell's connectivity.
Right.
This cell is connected this way, that there are photoreceptors in the eye that when light hits them, it excites the cell. There are photoreceptors around that area that when light hits them, it inhibits the cell. So in order to summarize that, you could show it with this circle.
Okay.
So you're summarizing the connectivity. Then I could give you, let's say, a light pattern. Now answering the question that came from this side. Then I could give you a light pattern and say, what do you think this light pattern gonna do with this, activity of this cell? Because now you don't have to look at the network because you have the receptive field.
Gotcha.
If you have the receptive field, you don't have to look at the network. You could say, well, in this case, it looks like there is light only in the excitatory area and there's no light in the inhibitory area. So this is an optimal stimulus for this cell. So the receptive field is summarizing what? The network. The network is being summarized in this format. It tells you how this cell gonna behave, right? You could look at the network, but the network gets complicated. So in order to not be dragged into what is all these little patterns are gonna do, you're gonna summarize it with the receptive field. They say, okay, this is the receptive field of this cell. It means that this cell responds to this pattern.
So it doesn't matter in that sense how strong is the connection. So you don't need to look into that.
Yeah.
To analyze.
You don't, no. So the receptive field summarizes some of those things for you.
But in the structure, how would you practice
so, well, let's look at it. If there is light in here, in these photoreceptors, what happens to the activity of this cell? Excites. What happens to the activity of this cell? What happens to the activity of this cell?
That's what I'm wondering, because it's both excited.
What do you mean? No. What happens to the activity of this cell? If there's light here. inhibits. So then there's a still, if I have another, 3 layers of neuron that are excitatory and eventually inhibitory, you still know that it's inhibitory surround. It doesn't matter. Like you just have to follow the network and then decide what is the impact of this light on this cell, right? The receptive field is summarizes the connectivity of a single cell. This cell has this receptive field. It's not just telling you that while this cell is sensitive to light in this part of the world, it tells you something more. It tells you that, well, the pattern of sensitivity is this.
Looking at the part on the left, I'm looking at the cell that's at the bottom left. Would it be possible for, I'm not saying this particular thing, but for the one that's coming down to inhibit this cell, but this cell would excite the one in the middle?
Yeah. Yeah, you could have a different pattern, right? You could have inhibitory, and then this would excite this. And you would still get an inhibitory surround, right? Because let's say this was a line, so inhibitory. Well, light here excites this, inhibits that, and because this was excitatory, inhibiting this, what the impact does it have on this one? What? Inhibitory. Because this cell, let's say, okay, this is not the pattern we're looking at anymore on the screen. So imagine this was inhibitory and this was excitatory. Well, if there's light here, it would excite this one, it would inhibit this one. If something that could have excited your cell itself is inhibited, the net effect is that it inhibits this cell. Does that make sense? We talked about this last time. Like you have to think about and be comfortable with these concepts. No, it means that you are detecting contrast in the world. So these cells, as we'll talk about in a few slides, can combine now. Let's say now there are a bunch of these cells next to each other, and another cell takes all these input. Then you could detect edges in the world because it's contrast, right? The world is all contrasts that we detect. And cells have to detect contrast in the world. Does that make sense? It's not like you're not detecting the light itself, you're detecting the contrast between these edges.
But the cell itself. Cell's not setting what you see, it's reacting.
The cells are representing, yes, of what is out there. So their activity signals what's out there. And in order for you to recognize, in order for you to have a visual perception, you have to, again, think about these as many layers. I have to come back to this. Every layer has a job, and then the next layer builds on top of that. The next layer builds on top of that. So, and then we'll get there at some point halfway through the course, hopefully, where we look at these networks. But you have to always think about, well, what one cell is not doing much, a layer is not doing much. It's just that across many layers, something emerges. Yeah. Okay, so as I said, if I change the connectivities here and I ask you what would be the receptive field, you should be comfortable with that. So you should know, well, why? Because you should be able to say, well, if I put light here, what happens to the activity of this cell? If I remove the light here, what happens to the activity of this cell, right? That way you should be able to draw based on the network connectivity. You have to try different patterns of light and then tell me what the receptive field would be. Because at that point, you should be able to tell, well, this part is excitatory, this part is inhibitory, excitatory, inhibitory. Well, this is the pattern of the receptive field. Questions?
Yeah, just to be super clear, so the reason that that's the receptive field, like that's the illustration of the cell network, is because the 2 on the outside are inhibitory and the 3 in the center are excitatory.
the light on the two on the outside will eventually have an inhibitory impact on this cell. The receptive field, the photoreceptors are not inhibitory. Again, it's just that the net effect of light in this area of the world is inhibitory on this cell. That's what it means. So, as I say, this is, if it wasn't like that, what kind of problem would you have here? If there wasn't an inhibitory surround, what would be the problem with this cell? There would be no contrast. It would not detect anything. If you had a small light or a bigger light or a bigger light or a whole sun, the cell would respond the same. And you don't want that. That's not interesting. A cell that responds to any pattern of light is not a useful cell. A cell that responds to a specific pattern of light in a specific part of the visual world, that's useful. What's optimal? The behavior? No, I'm just saying, if there was no inhibitory surround, if there were no contrast, then the cell would respond the same if you had a small light or a big light. So it doesn't really detect anything. But for any network or a receptive field, you could draw what the optimal, if I give you a whole network of a cell, it means that you know everything about this cell. Then you should be able to tell what patterns of light maximally activates the cell. And what would the pattern of light be? If you have a network, what pattern of light maximally activates the cell? No, what would be? So, well, there has to be light on photoreceptors that have a net excitatory effect on this cell, and it would be no light on photoreceptors that have a net inhibitory effect on this cell. In this case, this would be that. Well, there's light only in the area that is excitatory. There's no light when it's inhibitory. So this is the optimal stimulus. Yeah, you all follow that, right?
I'm curious, like, could you do an experiment on somebody who's colorblind and like their inhibitory and excitatory would look different than something that's more typical?
They're inhibitory.
Like, because I think colorblindness is something about contrast. I don't know. I'm just trying to think of practical ways for this too.
We're not going to talk about color perception that much, but there are networks like this that our people have figured out for color perception.
Okay.
And they show so it's a matter of certain photoreceptors not being in the eye usually.
Oh,
So certain photoreceptors, because again, we'll talk about this. Light is it's a rainbow. It's not just one thing. So there are a lot of different frequencies in light. And some of them if you can't detect some of them, you're not going to be able to perceive them. But it's not, we'll talk about it. So you should be able to know what the optimal stimulus is. And this, what we just saw, is basically centers around antagonistic organization. And this is a product of that lateral interaction, right? This sort of connectivity that you see here gives you that that centers around antagonistic organization. Again, try to draw different networks, try to see if you can figure out what the responses would be, what the optimal stimulus for them would be. Be comfortable with it. And also understand what does it mean to inhibit something that would have had excitatory thing, or excite something that would have had inhibitory thing, or not excite something that would have had inhibitory thing that much, right? That's another, you have to be comfortable with those things. As I said, some receptive fields have the opposite organization, inhibitory center, excitatory center. So the stuff that we talked about are not even in the visual cortex yet. They're even closer to the eye. But as you go into the visual cortex, you'll see patterns that are more complex. You see receptive fields that are more complex. And for that, we'll go back to this. Now, this is a cell in the visual cortex. Prior to this, as I said, it was in an area that LGN that we'll talk about later. But now it's not just a simple pattern of light, right? So that's what I mean. Networks build on top of each other, layers build on top of each other. You have to detect some little dots using that center surround antagonistic organization in order to now detect lines. So what is the area that they just mapped out? Excitatory or inhibitory? Excitatory. The light comes on, and you could see how selective the cell is, right? As soon as you go off, it doesn't fire anymore. But listen to this. So the light is on in this area, the light is on, and then what happens when it goes off? You hear it? So, no. So what happens when the light goes off? What happens? Yeah, so when the light is on, when the light is on, it's really quiet, right? But when light goes off, you hear it. What does that mean? It's in an inhibitory area. Because when you put light in this area, the cell goes quiet. You turn off the light. What is that like in our metaphor? When you turn the car metaphor. When the light goes off here, what are you doing? No, but you're picking your foot off the brake.
Wait, no, but you're slowing down, right?
No. So I'm talking when the light goes off in this area. So this is the exact moment I'm talking about. This moment. The moment that light went off in this area, the cell comes back to life a little, right? And that means that you're picking your foot off the brake pedal. This is an important moment. So let's listen to it again. This is a light. The light goes off, you hear a couple of spikes. Means that the car is kind of coming back to life because you pick your foot off the brake pedal. You could hear it still, right? When the light goes off in that area, you hear a little, you hear now strongly, right? You hear it more now. Here what happens when the light goes off here. You see? The light goes off. Why is that? Why is that? Why did it, when the light was narrow in this area, it went off, you hear a couple of spikes. Here, when the light is bigger, it goes off, you hear more spikes. Why? More cells being. So here, now it's a bigger inhibitory area was being activated. So when the light is bigger, a bigger inhibitory area was being activated. So when it goes off, the cells comes back to life is stronger. Does that make sense? This was a big inhibitory area. That cell has a big inhibitory area then. And when it goes off, the cell comes back to life. So they're not exciting the excitatory area at all. They're not touching the excitatory area. It's just that there was a big inhibitory area. It means that, there are a ton of brake pedals we're hitting, and then when you pick it up, the cell comes back to life quicker. And then as you move away from that area, it doesn't have that effect anymore. Why? No, it doesn't have any effect. It's outside the receptive field at that point. The receptive field includes the excitatory and inhibitory areas. If light goes on and off outside of that area, it doesn't have any impact. Right? You see that, right? When it's far away from that area, it doesn't matter. So they're mapping out the inhibitory area now. So you can see when the light moves in this direction, why don't you hear anything? Let's think about that. Why are we not hearing the self-fire here at all? Exactly, right? Or you have a little more inhibitory. When the light is like this, you have a little bit more inhibitory than excitatory. The excitatory is in the middle, but the inhibitory and the surround is really quieting the cell. So you see a cell could become really selective. But how do you get here from the receptive field we saw? Can you imagine how you get here? How does, let's say, as I said, this is a network. Every layer builds on prior layer. How would you get here? How do you get to this network from the prior center surround antagonist receptive field that we saw? Yeah, but how? If I put an excitatory center inhibitory surround receptive field that is here, a cell that has it here, another cell that has it here, another cell that has it here, another one, another one, another one. The aggregate effect that if there is another cell that gets excitatory input from all of these, what would be the response of that cell? Well, any light in the middle would excite it, any light on the outside would inhibit it. Does that make sense? Can you imagine a network? Okay, so this is how you get a so-called simple cell, yeah.
Is there a reason that they didn't go vertically or?
What do you mean vertically?
Well, vertically or diagonally, how did they know where to stop? Like they never shrunk down to know if this was going to continue down further.
Oh, I think they do that.
They eventually do.
Yeah, we could look at it later on. We'll come back to it. Yeah. So that's how you get a sort of more, these are called simple cells, and they are in the V1 in the first area in the visual cortex. I say that a lot, but we'll talk about it. And the receptive fields are now more elongated. They're not really simple anymore, like a circle. They're elongated. They look like this, and you saw how they look like in an experiment. They still have this antagonistic relationship because they're getting input from cells that are antagonistic, right? And those, when they aggregate, they give you these sorts of responses. So you could show in a schematic like this, where, well, when the light area is excitatory, and then the dark area is inhibitory. Or so this would be an on center off surround. This could be an off center on surround. And as you saw, those experimenters use this type of schematic, axis being excitatory and triangles being inhibitory. Okay, so, and as you saw, what does this do? If you have if you have a receptive field like this, what does it do? Well, it makes the cell really selective. The cell only responds to this specific orientation, because if it's orthogonal to that orientation, as you saw, the cell goes kind of quiet because you're hitting an equal number of excitatory and inhibitory stuff somewhat, a little more inhibitory. So the cell is not very responsive. But as soon as the orientation of the light matches the orientation that this specific cell likes, then what you see is that, well, the cell comes to life. They start firing really hard, saying, oh, I detected that there is an edge in the world, in this part of the world, with this angle, right? The cell is communicating that to the next layer. How does it know there is an edge there? Well, because it's listening, and these are metaphors, obviously. It's listening to cells that detects dots in that part of the world. And if enough of them are detected in a certain orientation, then this cell says, well, I detected an edge. So layers build on top of each other. So in this case, for example, if you had a certain orientation, the cell would be inhibited almost. A certain orientation, it would be baseline. A certain orientation that the cell would love. So they are tuned for this. They detect this. They respond to this. They like this, right? All of these things mean the same thing. It means that this specific pattern causes the cell to have a higher firing rate, right? That's what it means. But we use different languages to describe that. So look at this. If you light up everything, the cell is not going to respond. And that's a good thing. As I said, anything that responds to so if it's all light and the cell doesn't, respond to it, that's a good thing, right? Because it's not a meaningful pattern out there. You want cells that detects edges, that, as I said, so the edges become corners become shape of a chair, and then it builds on top of that. So you want your cells to be selective. And that's why that center surround antagonistic relationship is very important. But this is a behavior that you all expect, right? There is light only in the inhibitory area. And when it goes off, the cell is active. Because when there is some inhibitory, some excitatory, the cell is in the middle. Because as I said the last couple of weeks, every cell gets inhibitory stuff, gets excitatory stuff, and it combines them. It's just a matter of what is the ratio. If it's more excitatory, the cell would be above baseline. If it's more inhibitory, the cell would be below baseline. These people were very creative with their. All right, so we'll talk about complex cells after the exam. But these are what we call simple cells, because they're the simplest in the visual cortex. And what they do is they detect edges. They're the foundation of any visual experience. Edges. You have to have edges before you have anything else. But how are they constructed? So if now we have to think about in terms of networks. So you kind of now know what kind of networks give you those circular center surround receptive field. Now you have to think about how could those responses be combined in order to give you these elongated center surround responses, right? So let's say this is an example. This is one way to think about it. Again, it's in a schematic just for you to be able to imagine this yourself, how this would play out in a network, right? If I have a bunch of different cells. Now, as I said, a cell, when we're talking about the cells that we talked about, these are in an area before the visual cortex, right? The visual input leaves the eye, it makes a stop in LGN. After that stop, it goes to the visual cortex. So you could look at the LGN responses, and these responses are from LGN. Now, you could imagine that when I say, well, this cell likes this orientation in this part of the world, well, there's another cell that likes that orientation in different part of the world. There's another cell that likes another orientation in this part of the world, right? It's blanketed. Like there are so many different cells in the visual cortex, in the LGN, there are so many cells in the retina, and they basically blanket this space of possibilities. For every orientation, every location in this space, there is a cell that detects that. There are a million cells that respond to that, right? There are a lot of cells that respond to every combination of things. So you could imagine there are cells that respond to light in this part of the world. There are cells that respond to light next to it, respond to light next to it, next to it, next to it. Now, if on the next layer there is a cell that is connected to these ones, then it's conceivable that it would eventually emerge to have responses that we just saw. Because if all the cells in the prior layer have these center surround antagonistic relationship, then if you put light in the middle, it's likely to excite an excitatory area of these cells. And in turn, this would excite the simple cell that we just saw. If there's light on the outside area, it's likely that it would inhibit these cells, and these cells have excitatory connection. So in turn, it would inhibit this cell. Does that make sense? So you can imagine a network on the next layer where these sorts of selectivities could emerge. Questions? Yeah, this is just, we're not going to draw these networks, but I want you to have a mental model of these networks, right? These networks are just, they're basically cells that are connected, and depending on the pattern of light and the connectivity that they have, they'll have a certain behavior. Behavior for a cell means whether it would fire or not. Like it's not like there's, there isn't much else it can do, as I said. So think of the brain as this network that has units. These units are connected, they impact one another. So there's connectivity. And the unit could have activity, but the activity is a function of that connectivity and the input. It's not by itself. You cannot tell me what the activity of the cell is just by the network or just by the receptive field. I have to give you that light, the input too, right? You can't predict the response of a cell unless you know what the person is looking at, what the monkey or the cat in this case is looking at. So you have to know the visual input, you have to know the network, and then you could tell me what the response of this cell would be. Right? So think in terms of activation patterns and things in terms of connectivity. Those are the two things that goes on in the brain. And layers. Layers and hierarchy gives you complexity, gives you a process to build on top of each other. And you could see this in nature, you could see this in the brain, right? Hierarchy gives us complexity, like you could build on top of each other. Yeah.
Speaking on the layers, does each individual photoreceptor, is it associated with one receptive field, or are photoreceptors have like multiple connections going to
Yeah, they could be connected to thousands of cells, right? One photoreceptor could be connected to many cells, right? It just overlaps, yeah. And those are connected to many cells and many cells. So it's everything's shared in the system, yeah. But as I said, in the foveal or central vision, that's what we call, photoreceptors are not connected to too many cells because you want granularity. And we talk about what that means later on, because you could even see in these networks, right? That if you're getting a lot of inputs, many different patterns of input gives you the same neural activity. So you cannot really dissociate between them. But if you get a limited number of inputs, you're limited to how many patterns you could really see. So it gives you precision. All right. So these elongated receptive fields are constructed by combining the receptive fields of simpler cells. And they really function as feature detectors. You could think of them as this cell's job, based on its connectivity, is to detect an edge somewhere. And another cell's job is to detect an edge somewhere else, next to it, or next to it, or next to it. And then down the line, there is a cell that puts all of those together and detects a corner. Then after that, there's a cell that puts all of those together, detects a shape, maybe of a face or something. So, so network layer by layer build these complexity. And that's a metaphor again. Feature detection is a metaphor. Representation is another metaphor. It's representing this or representing that. I think that and again, another word that I have here is tuning, right? You could say this cell is tuned for this orientation, which means that this orientation maximally activates this cell. This cell detects that orientation. This cell responds to that orientation. This cell represents that orientation. These are all the same thing. Just different way of describing the very similar behavior that this cell would have. Questions?
For saccades or something, is that a lack of photoreceptors to a specific receptive field, or is it like multiple chains that are just inventory?
Saccades, the eye movements are to bring the high resolution fovea to a location, right? So do this experiment, right? Look at this, look at where my mouse is. Don't move your eye and try to read the sentence, right? How far do you get? One word, maybe, right? If you just keep your fixation here and try to read the sentence. I'll get up to this point, maybe myself. You get all that, right? Why is that? Well, because reading requires very high resolution. Letters are very similar to one another. And that kind of resolution, you could only have that with low convergence. And you could only have that in the central fixation. So you have to move your eye to the next word, to the next word, to the next word. So the word actually falls on your like we'll talk about it, on the area that has low convergence. And if it has low convergence, the responses don't mix up, and then you're able to recognize them. But the reason we make eye movements is because of that trade-off between central vision and peripheral vision. Any questions? You're all confident with this, and I'm sure the TAs will help you understand this, but as I said, you should all study before coming to that session on Friday. You should all study, you should all find what your weakest spots are, and try to come with your question. Come trying to get the most out of that, not coming just trying to listen in again, like just to what they say or something like that. Try to study, and then I'm sure you're all gonna do well. All right, see you next week.